Ha. A warm namaste and welcome to all. I, Snehal Patil, heartily welcome you. National webinar organized by Mula Education Society, Arts, Commerce, Science College, Sonai, Ahmednagar, in association with Sri Shivaji Education Society's Yashwantrao Chavan College of Science, Karad, District Satara, on the occasion of bicentennial celebration of Brigadier Mendel. I welcome to Honorable Speaker Dr. B.J. Apparao, Director Collegiate Education, Mula Education Society, Nevasa, Ahmednagar, and also Dr. Rupesh Bedre sir, Associate Professor, Department of Botany, Sant Tukdoji Maharaj University, Nagpur, and also President of today's program, Dr. S.H. Burungre sir, Professor Yashwantrao Chavan College of Science, Karad, Chemistry Department, and all those who are joining us in this online webinar. I request to Dr. S.A. Kirtane Madam to deliver the introduction and as well as welcome address.
Thank you, Madam. First of all, I tribute to the great personalities not only of India but the world also. Chhatrapati Shivaji Maharaj, Mahatma Jyotiba Phule, Savitribai Phule, Rajashri Shahu Maharaj, Dr. Babasaheb Ambedkar, Swargiya Yashwantraoji Chauhan Saheb, and all who have devoted their lives for betterment of society. Honorable President Dr. S.H. Burungale sir, today's Honorable Speaker Dr. B.J. Apparao sir, Director Collegiate Education, Mula Education Society, Nevasa, Ahmednagar, and Dr. Rupesh Bedare, Associate Professor, Department of Botany, Sant Tukdoji Maharaj, Nagpur University, Nagpur. Honorable Principal Dr. S.B. Kengar sir, Honorable Principal Dr. S.L. Laure sir, Dr. Podda sir, and all my colleagues and you all respected participants. I feel extremely privileged and honored to be given this opportunity to welcome you all. On behalf of our college, I welcome you all and thank you for taking the time to join us today. We are using online platform from the year 2020 because of COVID-19. The whole world has been profoundly affected by global virus pandemic, but in this situation also, teaching learning process is continued very effectively because of this online platform. One important plus point of this online platform is participants can join from various localities, can share their ideas, and can discuss on various aspects for improvement of knowledge. Distance doesn't matter. Today, we all are under same umbrella for bicentennial celebration of the great genius of genetics, father of genetics, Gregor John Mendel. He discovered the basic rules of inheritance. The transmission of genes follows the same laws that Mendel had proposed in 1866. I tribute to Gregor John Mendel on his birth bicentennial. We salute him as a paragon of science. I warmly welcome you again and thankful to our President, Honorable Speakers, dear participants, for sparing valuable time with us to share some ideas and grace the event. Jai Hind, Jai Maharashtra, Jai Bhim. Thank you. Thank you.
Thank you, ma'am. Then I request Principal Dr. S.L. Laure sir for convenor speech.
Good afternoon. Today we organized a national level webinar on the occasion of bicentenary anniversary of Mendel, who has discovered a factor. Now it is called gene and DNA. There are many more things. Today we have with us Dr. B.J. Apparao, Director Collegiate Education, Mula Education Society, Nevasa, as a speaker, as well as Honorable Dr. Rupesh Bedare, Associate Professor, Department of Botany, STM Nagpur University, Nagpur. My fellow colleagues and the Principal of Yashwantrao Chavan College of Science, Dr. Kengar sir, Dr. Podda sir, Dr. S.H. Burungale sir, Sushma Kirtane, Organizing Secretary, my colleagues from my department, and our IQAC coordinator, Dr. Sandeep Khedkar, sir. I welcome you all. This bicentenary anniversary function we are going to organize today. This gives an idea how Mendel has worked and what are the contributions. So definitely we'll learn many more things out of these two lectures. Thank you.
Thank you, sir. Then I invite Dr. S.H. Burungre, sir, Professor Yashwantrao Chavan, College of Science, Karad, for presidential address.
Thank you, madam. आजच्या या राष्ट्रीय योजनाच्या निमित्ताने आज मेंडेले यांचा 200वी वाढदिवस साजरा करण्याचा जो संकल्प दोन्ही महाविद्यालयाने घेतला आहे तो प्रत्येक सजीवामधील एक बदल आहे. सन्मान्य माननीय प्राचार्य डॉक्टर एस.एल. लावारे सर, सन्मान्य डॉक्टर सचिन खेडकर सर, अप्पाराव सर, रुपेश बेद्रे आणि वनस्पती विभागाचे विभाग प्रमुख पोद्दार सर आणि त्यांचे सहकारी कीर्तनी मॅडम, मला या कार्यक्रमाच्या निमित्तानं थोडक्यात मी माझं मनोगत व्यक्त करत आहे. खरं तर या 19व्या शतकात सामान्यतः असे मानले जाते की प्रत्येक पालकाने दान केलेले जीवाचे गुणधर्म पुढील संततीमध्ये दिले जातात. परंतु त्यावेळी किंवा त्या काळात अनुवंशिकता समजली गेली नाही आणि या जनुकांची जी काय संकल्पना मुळीच अस्तित्वात नव्हते. परंतु त्या काळी ऑस्ट्रेलियामध्ये जन्माला आलेल्या ग्रेगोर मेंडेले यांनी जनुकाचा शोध लावला आणि त्यानंतर त्याचे शोध प्रकाशित झाले नाही. अवघे या मेंडेले यांना फक्त 62 वर्षाचाच कारण त्यांचा जन्मच झाला होता की 20 जुलै 1822 ते 6 जानेवारी 1884. या अवघ्या 62व्या वर्षी त्यांचं निधन झालं आणि नंतरच्या काळात काही युरोपियन शास्त्रज्ञांनी या अणुशास्त्राच्या आधुनिक विज्ञानाचे संस्थापक म्हणून मरणोत्तर मान्यता प्राप्त झाली. त्याने असे लिहून ठेवले आहे की माझ्या वैज्ञानिक कार्यामुळं मला इतके समाधान मिळाले की मला खात्री आहे की संपूर्ण जग या अभ्यासाचे परिणाम ओळखेल आणि अवघ्या ह्या जो संक्रमण आहे, जो बदल आहे, मागील पिढीपासून पुढील पिढीपर्यंत जो बदल होणार आहे तो हे नंतरच्या शास्त्रज्ञाने एक गणितीय पद्धतीने हे सूत्र मांडण्यात आलं आणि तो जो बदल आहे तो मुळात त्या मेंडेलियनचा होता हे त्यावेळी या 3 शास्त्रज्ञांनी दाखवून दिलं आणि जगाला कळालं की खरं हे जेनेटिक्सचा जो फादर आहे तो मेंडेले आहे. या व्याख्यानातून मला निश्चित वाटतं की जे मान्यवर आहेत, सचिन खेडेकर असतील, अप्पाराव सर असतील, रुपेश बेद्रे सर असतील, यांच्या व्याख्यानातून निश्चित आपल्या सर्वांपर्यंत एक चांगला संदेश, चांगलं मार्गदर्शन मिळेल अशी आशा व्यक्त करतो आणि माझे 2 शब्द थांबवतो. धन्यवाद.
Thank you, sir. Then I request Dr. Sandeep Khedkar, sir, to introduce today's honorable speaker, Dr. B.J. Apparao, sir.
Thank you, madam. Good afternoon, everybody. I take this as an honor to introduce today's chief guest, Honorable Dr. B.J. Apparao, sir. Dr. B.J. Apparao, sir, previously worked as a principal at Jizamata College, Mendel. At present, he is functioning as Director of Research of Collegiate Education of Mula Education Society, Sonai. To honor Gregor Mendel and other biologists, I would say that he is the great origin. Our principal, Dr. B.J. Apparao sir, is a great origin of species of for us as an academician. To his credit, there are 14 scholars who have completed their PhD under his guidance. He has published 65 research papers in reputed national and international journals, and also co-authored 8 books in reputed publications. So, with this, I welcome Honorable Dr. B.J. Apparao sir. Thank you.
Thank you, sir. Then I request to Ghadge ma'am to introduce Honorable Speaker Dr. Rupesh Bedre, sir.
Thank you, ma'am. Let me take this opportunity to introduce you to our next eminent guest on today's program, Dr. Rupesh Bedre, sir. He Associate Professor, Department of Botany, Rashtrasant Tukroji Maharaj, Nagpur University, Nagpur. He completed his PhD from Rashtrasant Tukroji Maharaj, Nagpur University, Nagpur, on subject investigation on introduced mutations in viral NL197 and RLC6 of licensed species for quantitative and qualitative improvement. He has been awarded by a fellowship of Indian Botanical Society, postdoctoral fellowship, Department of Biotechnology, Government of India in 2003 to 2005. School of Life Sciences, Jawaharlal Nehru University, India. He has successfully completed 5 major research projects funded by UGC, BRNS, DAE, and RTM, Nagpur University, Nagpur. Further, to acknowledge his area of specialization and interest, his research interests have been in the area of introduced mutagens in plants and the plants area, plant defense, organic bioscience, and the plant tissues culture. He is an active PhD guide at Rashtrasant Tukroji Maharaj University, Nagpur. Currently, he is guided PhD students for the scholars. He has attended various conferences, national and international. He has delivered the lectures on scientific temperament and the social awareness. He has also published more than 45 research papers in national and international journals and have 112 citations and one book chapter published, one textbook published. I would like to our knowledge hand over to stage our guest to share this knowledge to our participants. Please, sir.
Thank you, ma'am. When I request to Honorable Speaker Dr. B.J. Apparao, sir, to address us on the topic Mendel, father of genetics.
Especially Dr. Bargule, sir, president of this function, Dr. S.B. Kenger, principal as well as chairman for this session, convener, Dr. Shankar Lavare, sir, organizing secretary, Dr. Sushma Kirtane, Dr. Rupesh Bedre, sir, and all participants of this session and dear friends. It gives me pleasure to be with you this afternoon and discussing about my God, my favorite guru, John Gregor Mendel. Since I was young, maybe in BSc, since then I was very much influenced by, very much interested by Mendelian genetics, and Ame were a disciple of genetics. Johann Gregor Mendel actually is not a scientist. He didn't get a PhD degree, but as a monk, he was an Austrian monk rather. I think today I will be sharing with you some information about the genius of genetics or father of genetics, Johann Gregor Mendel. All of you know we have completed around 200 years and bicentenary celebrations we are celebrating. And Gregor Johann Mendel, that was his full name, born on this day, 22nd July, 1822, and he died at the age of 62, that is on 6th January, 1884. He belonged to the earlier Brunn, Austria. I think now it is in Czechoslovakia. Czechoslovakia is again subdivided into Czech Republic and Slovakia, and this place comes under Slovakia, Brunn. So actually, he worked as a priest. He was born to two uneducated parents whose occupation is farming in Austria, Brunn. So he grew up, his childhood education was done in the Brunn itself. And at the age of 23, he went to, at the age of 18, he was appointed as a priest for that monastery at Vienna. Then after that, at the age of 23, he was sent to Vienna University, Austria. So there he selected subjects like mathematics, physics, and natural sciences. And with that, he has stopped his education. He completed a university education, came back to the monastery and worked as a priest as well as a school teacher also. So main career was like a school teacher. So here occupation worked as a priest or monk and a teacher in high school. He was very much impressed with plant breeding experiments at that time. So Kohlreuter was his teacher at Vienna University in natural sciences. He gained some interest from Kohlreuter, and another Nageli was one of the botanists. He was there, and from these two, he got very much interest, and he started plant breeding experiments in Pisum sativum P. He has a very small garden, a small space in the monastery where he did all his experiments. So the name of the experimental plant material is used is P. Pisum sativum, which belongs to the family Papilionaceae, and it has chromosome number N is 7 or 2N is equal to 14. So in monastery garden itself, he carried out all this work. That was between 1857 and 1863. He cultivated something like 29,000 plants he has tested. Mendel did not, whatever he have done, it is just by luck. He has got very good God's grace. That is why he became a father of genetics. No, he carefully selected everything, calculated scientifically, whatever the knowledge he has got. So with that, he selected pea plants first. Why? Pea plant is a self-pollinated plant. Cross-pollination does not occur. Whatever the hybridization takes place, the characters remain as pure inside. Then second, luckily he has selected 7 different characters unknowingly, which are present on 7 different chromosomes. Here you can see length of the stem he has taken. Even though length of the stem is a quantitative trait, he did not measure the length, but only qualitatively, whether it is looking tall or looking dwarf, such criteria is taken. Position of the flowers, whether they are axial or terminal, color of the flower, whether it is purple or white, all these are qualitative traits. Actually, Mendel did not select any quantitative trait, quantitative character. So all these 7 qualitative traits he has selected, and luckily later we came to know all these 7 characters were present on 7 different chromosomes. 7 different chromosomes. One chromosome has 1 character. So he was not knowing about the linkage. The linkage concept has come later. So there was no linkage. There was no quantitative traits, characters. Quantitative traits are controlled by several genes, several alleles, etc. In these 2 aspects, he was lucky. But otherwise, very studiedly, he has selected all these 7 characters. So here again, I repeat the sentence. What Mendel did with P was not an accident, but was the result of his careful thoughts and calculations. Even though he's not a scientist, he chose proper plant. As I told you, he chose pea plants whose life cycle is very short. Plants are self-pollinated and pure. Second, he took 1 character at a time in his experiments of hybridization. Suppose purple petal color, flower color, crossed with white color. What will happen? Generation. He has analyzed all this. After 1 cross is over, he recorded all his observation very carefully, and then he took up second experiment, maybe tall versus dwarf, like that. When monohybrid cross is over, means a hybridization cross taking single character is over, then he has taken dihybrid crosses. So like that, step by step he has taken, that is another great thing. He didn't mess up the things. Things were very crystal clear to him. He was lucky all 7 characters he chose are regulated by a single gene that are found on different chromosomes. I think that I've already explained to you. Hybridization experiments were easy to carry out on pea plant, and Mendel only performed a dihybrid cross after completion of the monohybrid cross. So he only shows those characters that were contrasting. Suppose petal color, red, pink is there, then what is the contrasting color? White color. Now we know, after the development of the science and all, if a dominant gene, some mutation occurs, it loses its character, it becomes white. So such 7 characters he has selected. So it was at University of Vienna, Mendel picked up knowledge about Kohlreuter's and Gardner's hybridization experiments. So after completing university education, Mendel returned to Brno in 1854. After 3 years, he continued, from 84 onwards, he continued to work as a priest and as a teacher in high school. And only in 1856, in some papers, they have mentioned 57. He began his famous experiments on peace in the monastery garden. 7 years it took, means from 1856 to 63, 7 years, he has strived, worked hard, doing hybridization experiments in pea systematically, recording everything. Nothing he left to choice. Everything he has recorded. And as many as 29,000 plants he has analyzed during these 7 years and odd. So Mendel was able to describe, based on their observations, clear analysis, 3 laws he has put up. They are one, law of segregation, law of independent assortment, and the law of dominance is the first one we say. But law of dominance, law of segregation, and law of independent assortment. On March 8, 1965, Gregor Mendel presented his findings, experiments in plant hybridization at the Natural History Society of Brno. It was published a year later in the annual proceedings of Natural History Society in 1866. This great work of Mendel did not attract the slightest attention from scientific community. His publication went unrecognized and Mendel got bitterly disappointed. Mendel died in the year 1884 at the age of 62. After around 34 years, the Mendelism was rediscovered. Three great scientists, Hugo de Vries, who is a Dutch botanist, Paul Correns, a German botanist, and one Schermark, Austrian botanist. Individually, they belong to different countries and they were working on hereditary. And as you know, as conventionally, if you are working anything for your PhD program or anything, we go for a literature survey. While surveying the literature, surveying the literature, they found some person by name Gregor John Mendel, 34 years ago. He has worked a very good piece of work in a plant and he has given his ideas, his work, proposed three theories. And surprisingly, these three scientists, three botanists, they did not claim it is their original, but the person who has carried out the research 34 years ago was given the credit. Mendel, he discovered already. We were just rediscovering what he discovered 34 years ago. So, as I told you, Mendel, based on his experiments, proposed three laws of heredity. First one he called law of dominance. Law of dominance. Whatever the character a plant is getting, the character, I mean, by what do we mean by characters? The characters are the one which you see, phenotypic character, the characters which you can see with your naked eyes. So genotype, that is a different thing, all of you know that. So phenotypic characters he has taken. As I already explained, the 7 different characters he has selected. So I think in this picture I have shown, it's a dihybrid cross, yellow color seed, that is one character. Its contrasting character is green color seed. When you talk about character, color, seed color is yellow, its contrasting color is green. The second character, shape of the seed, whether the seed is round or wrinkled. So here in this diagram, whatever you can see here, this one, it has gone. Yes, you can see this one is two characters. You can see one is, it is yellow in color and round in shape. The second one is wrinkled in shape but green in color. So these are the contrasting characters he has taken and he crossed it and tried to observe after hybridization the product we call first filial generation. First generation, what will be the character? Suppose first generation, you are getting this traits only. Means why yellow character, yellow color seed is given in the first generation, he thought that even though capital or small r, both the genes are there in that, both the factors, he called it as factors. Yellow color factor is dominant over the small y. So he called it law of dominance. Both the factors, dominant factor and a recessive factor, both are coexisting in the same cell. But one trait, one character, one factor expresses itself while the other character or other factor remains silent. He called it as law of dominance. So in the words we explain, in general terms, we can say the ability of one allele to express its phenotype. Generally, the dominant allele will make the gene product that the recessive cannot. This is the modern explanation. Therefore, the dominant allele will express itself whenever it is present. So that we know, that is the present knowledge. Mendel was not knowing at that time, even not even chromosomes were not well established. DNA was not known. Genes, there was no question about it. So using a factor, he said that there is some factor which is dominant. So it is giving that character in the first filial generation also. Like that, law of segregation, second. He believed that when a plant, a parent is producing its gametes, all of you know that meiosis occurs. At meiosis, the genes will segregate. Genes will segregate. So suppose capital T, small t are together in the parent, but at the time of gamete formation, the capital T or the dominant factor and the small t, that is the recessive factor, both will be segregated and they go to different gametes. Suppose pollen grains, both will go to one pollen grain may contain capital T, another pollen grain will contain small t. So that he called a law of segregation. Similarly, same thing happens in the female parent also. Female parent gives the female gametes egg cells. Some egg cells will get capital T, some egg cells may get small t, recessive factor. When during the hybridization, one male gamete containing either capital T or small t can combine with an egg cell containing either capital T or small t. Together, it will give either capital T or capital T small t or small t. So that way, the segregation occurs independent. So gametes, they can mix with each other independently. Any male gamete can combine with any female gamete. Similarly, any female gamete has a chance of getting mated with any of the male gametes. So he called it as law of independent mixing. Assortment means it is mixing. Law of independent mixing. So here, these are the three laws Mendel has given. Anyway, we are not going deep into the subject. I want to give a brief information about the Mendel's life, etc. Next one. He did monohybrid crosses and dihybrid crosses. Monohybrid cross means mono, single, hybrid cross. So here only one character at a time was studied. So here flower you can see a purple plant, purple color flowered plant is crossed with a white flower producing parent. So when purple and white was crossed in first filial generation, all were purple. This indicates the law of dominance. This indicates law of dominance. But when these F1 filial progeny were intercrossed with each other or self, Again, the character which is disappearing in the F1 generation, that is white trait, that started reappearing here in the second one. Here you can see actually this white factor was disappearing in first filial generation. It reappeared in a 3:1 ratio. This not once, not twice, several, maybe hundreds of times he has repeated, and plants were changing. I mean, the characters were changing, but the result is the same. He repeated all 7 characters, result is the same. When you concentrate on monohybrid cross, it was always 3:1, 3 dominants to 1 recessive. Similarly, I think he has given, he tried to give explanation here. Maybe purple color is producing because of 2 dominant alleles. We know this pea plant is a diploid plant. Diploid. Homologous chromosomes will be there. Each chromosome is having, and the parents are pure. Parents are pure. So small capital P we have. Similarly, the recessive plant is always white. There is no purple color pigment producing gene here. So it is unable to produce purple color. It is producing only white color. In absence of capital P, it produces white petal color. So he said that law of segregation, these two throughout the life of the plant, but they are together. But they, at the time of gamete formation, capital P goes to one gamete and another gamete. Suppose if it is female, then it is a female gamete, another female gamete. So here, the white is a male parent, suppose, then small p and small p is there. Now, this p has got equal chances of combining with any of the male gametes. Similarly, this p has got equal chances of combining with any of the male gametes. And so, same thing here. Male gamete, either it can pollinate this or this. So random chances are there. If you do it anyway, you will get one capital P and small p. We call it as heterozygote. These are homozygotes, pure plants. But this first filial generation has got heterozygotes. One capital P, one small p. All are heterozygotes. Since P is dominant, they will produce purple only. All 4 purple plant. When you self these 2 plants, self cross, or any 2, if you self it, again the same. The capital P and small p, they separate, and they go to different gametes and all. Independent assortment will be there. This capital P can combine either with this or with this. If it is combining with this, it will give capital P, or if it is combining with this, it will give capital P, small p, and so on. And then you see this capital P will be purple just like this. This will be the purple. So you will have plants produced in 3:1, 300 plants. Suppose there are 400 plants, 300 plants plus or minus will be purple, 100 and all plants will be white. This is how John Gregor Mendel performed so many experiments, and he got very crystal clear results which he analyzed, even though he is not a scientist, even though he does not have a scientific background. During all his experiments, John Gregor Mendel used to go to Vienna University. In Vienna University, he used to meet one very well-known, renowned botanist is there. His name is Nageli. He used to discuss with him everything, even while writing the paper, publishing it in Proceedings of National Natural History Museum. And then in all this, he was there, but even Dr. Nageli failed to appreciate his work. They could not realize the importance of this work. Similarly, Mendel also has done dihybrid crosses and test crosses, then back crosses and all. Even dihybrid cross later on it was known, and some ratios I have given. These ratios, they indicate, all these they indicate that the factors, the so-called genes, they are inherited from the parents to the offspring, and that factor is responsible for the producing that trait. So that indicates, that has given some idea why always human beings produce human beings with the same character. Why not cats or buffaloes? Why a pea plant produces always pea plants? Why not roses or anything? Because these factors, they inherit, they are transmitted from parents to offspring, and they control the characters. Why Mendel was called father of genetics? Mendel was the first to give scientific explanation regarding the mode of transmission of characters and formulate the basic laws of heredity. So he is rightly called the father of genetics. He is also considered to be the founder of modern genetics. The pity is that at various times, John Gregor Mendel has been honored. Those who have recognized his work, they honored him. Sometimes his work is dismissed. Sometimes his work is always rediscovered after 34 years. Some people doubted it, but in spite of all this, his important contributions to the field of genetics continues to be proved to be correct even today. During these 200 years, many people are working in genetics, working with different plant materials, different things. We know what are mutations, we know genetical explanation, we know DNA, genes, and everything. Everything, just whatever Mendel thought, in the same thought still prevails. Only terminology is changing. We say chromosomes, DNA, genes, etc. 200 years of Gregor John Mendel, although unrecognized for his scientific achievements during his life, Gregor Mendel pioneered our modern understanding of the gene, work that shaped the field of genetics. So I would salute the great personality ever walked on this earth, even though he is not a scientist, with his little education and all, but great scientific spirits. He has discovered, he has changed the world, changed our understanding about the heredity. I had an opportunity to visit his garden. You can see here, this is the exact place where John Gregor Mendel has conducted his experiments with pea. And this is the monastery, and this is the museum now they are running here. And there is a big statue of John Gregor Mendel here. So dear friends, even though it is preliminary or anything, I just want to provoke interest about this Mendelism in the younger minds, and I thank the organizers to give me for giving me this opportunity, and thank you all for your patience. Listen, thank you.
Thank you, sir. Then I request to Honorable Speaker Dr. Rupesh Bedre, sir, on topic genetics beyond Mendel.
Good evening. Am I audible? Hello.
Yes,
Ah, yes,
yes,
sir.
sir. Yes, sir.
Good evening. First of all, I right. I want to thank Mula Education Society and Arts, Commerce and Science College, the principal and the staff of Arts, Commerce and Science College, Sonai, as well as the principal and staff of Yashwantrao Chavan College of Science, Karad. And my special thank to Kirtane Madam for inviting me, for giving me an opportunity to speak to you and your students specifically on this occasion of bicentennial birth anniversary of Mendel, John Gregor Mendel, whom we know as a father of genetics. Just now, Dr. Apparao has, in a very lucid manner, in very detail, has explained about the experiments of Mendel carried out by Mendel, as well as he has also shed a light on the life of Mendel and how he was brought up, how he was trained, and what was his profession. And specifically, he has highlighted the interest of Mendel, which led to the discovery of laws of inheritance. So, with the permission of chairman, may I start my presentation? Hello. Hello.
Yes, sir.
Am I? May I start?
Yes, sir.
Yes, sir.
Okay. So I hope my screen is visible to all, right?
Yes, it's visible.
Yeah. Just now, Dr. Apparao has in very detail, much detail, explained about the Mendel, his life history as well as the scientific experiments. But the sad part of it was that his work was not recognized in his lifetime. Although after his death, he was enshrined, he was given the title of father of genetics. But when he was alive, his work was not recognized. There are various reasons because behind that. As Dr. Apparao has said that he has presented his work in 65 and 66. And the whole presentation was wrongly understood by the audience. There were around 40 odd audience sitting in that room, and none of those was able to understand that what Mendel is speaking is about inheritance. He is not speaking about hybridization. And his some innovative ways of dealing the subject led to the ignorance and criticism in his lifetime. But Mendel was not disheartened. It is said that he used to say to his friend, which I have written over here, my time will come. Yes, it came, but the sad part was that it came after his death. He was unable to witness himself the fruits of his work. That is a bad part. But as it is called in Hindi, Der Aaye Durust Aaye, some 14 years after his death, his work was rediscovered by 3 honest scientists, and the whole credit was given to him. And since uh, Dr. Apparao has already explained everything about Mendel, I am not going in detail about that. Although I had prepared some slides, but he has gone in much detail. It will be just repetition. So I will directly come to my topic. That, oh sorry. as soon as the Mendel's work was discovered, people tried to do the similar experiments, and most of time they were unable to get the similar results which Mendel got not in peas, in other plants, in other organisms also, not only plants. And there were several reasons behind that. As Dr. Apparao has said that whatever he chose were the characters which were monogenic. But there are many characters which are not monogenic. They are under the control of more than one gene. So obviously they are not going to follow the laws of given by Mendel. Then in case of Mendel, there was clear-cut dominant recessive relationship between the traits, but that is not always there. And again, a character may be influenced by more than one gene, that is there. Similarly, the two characters may be linked. The genes controlling two characters, they may be linked, which was not the case in case of Mendel. So due to all those reasons, people were unable to always get the results which followed the Mendel's laws of inheritance, as we call it now. And they started working on that in detail. And they found that the results vary from Mendel with respect to all these 5 major classes. There are allelic interactions, then there is penetrance and expressivity for the genes, then there are multiple alleles, there are isoalleles, then characters are under the control of more than one gene, and those genes, they interact in various ways. And then of course the linkage, the two genes, they are linked, which control two different characters, and that's why they will always not behave Mendel's laws of independent assortment. So we shall see one by one. Now, the first is the allelic interaction, that is the incomplete dominance and the codominance. The genes, they have alternate forms, which we call them as alleles. Alleles are nothing but the nucleotide variants of a gene. The sequence of nucleotide varies in those genes, and we call them as alleles. In case of Mendel, there was strict dominant recessive relationship, but this is not always. There can be incomplete dominance. An allele may not be completely dominant over some other allele, and that's why it is not completely expressed, like in case of Mirabilis jalapa. There are red flowers, there are white plants having white flowers. If you cross them, according to Mendel, either it should have been red or it should have been white. But the F1 is pink in color, which is intermediate of red and white. That is because the allele which codes for redness, it is incompletely dominant over the allele which controls the white character. Then another is codominance, where both the alleles in an heterozygous individual are equally expressed. So as you can see in this image, the progeny expresses the character of male parent as well as the female parent, and this is very well what you can say expressed in form of ABO blood groups. The blood group is under the control of gene I, which has 3 alleles, IA, IB, and IO. IA as well as IB, they are dominant over IO. Means IO is recessive. But IA and IB, they are codominant. And that's why the individual which has the genotype IA, IB, it has the blood group AB. Homozygous for IO, it has the blood group of O. Then others, there are A or B blood group depending upon the genetic constitution. So in this case, you see that the allele IA is also expressed, allele IB is also expressed. They are codominant. But this dominant recessive relationship is not absolute. It is relative. It depends on the level in which you are speaking. So phenotype is at morphological level, which we can see, the form of an organism, which we can see with our eyes. Then there is a physiological phenotype, and then there is a molecular phenotype, which is expressed inside the cell. And this you can understand by taking the example of cystic fibrosis. It is a disorder in which the air ducts in the lungs, they are blocked. The patient suffering from cystic fibrosis, the air ducts, they produce large amount of mucus, thick, sticky mucus, which clogs those air ducts. Now, this phenotype of producing mucus is under the control of gene CFTR, that is cystic fibrosis transmembrane conductance regulator. And this gene is present on the long arm of chromosome number 7. The wild type will code the proper protein, it will function properly, and the patient will be healthy. But if it has homozygous mutated gene, then the person will suffer from cystic fibrosis. So this CFTR gene, what it does, it codes for a transmembrane protein, the intrinsic protein, right? It is a transporter of chloride ions. The normally functioning transporter coded by CFTR will pump out chloride ions, excess of chloride ions from cell into the out of the cell. But if the mutated allele is there, it will code for a dysfunctional transporter, which is unable to pump out the excess chloride ions. And that's why the chloride ions, they accumulate inside the cell. So then the question arises whether it is recessive or it is codominant. When the CFTR is homozygous, wild type homozygous, then the person is normal. If it is mutated homozygous, then the person is affected by cystic fibrosis. If the person is heterozygous, then again it is normal. The person is normal. He won't carry that. He won't have that disease. He won't suffer from that disease. So this clearly tells that it is a case of recessive inheritance. The mutated CFTR is recessive to the wild type of dominant gene, dominant allele. But if you go at the molecular level, then it is codominant. Why? Because in heterozygous individual, the wild type allele will also be expressed. It will produce its protein. The mutated allele will also be expressed. It will also produce the protein. So obviously, both the alleles are being expressed. Their molecular phenotype is there. Then it is a case of codominance. Although in that heterozygous individual, the functional CFTR protein will take care of chloride ions, and the person will not be suffering from the disease. But at physiological level, the disease is recessive. At molecular level, it is codominant. So we have to be very much careful about this. While saying that a character is codominant or recessive or incompletely dominant, you also you have to always keep in mind at what level you are speaking, whether you are speaking at molecular level or physiological level or at the morphological level. Then next most important is penetrance and expressivity. In case of Mendel, okay, before that, let me touch upon this thing. A very important formula, you can say. The phenotype, what we see in an individual, it is the manifestation of the interaction between the genotype of the individual and the environment. So this equation, what you can tell about the limitation of the genotype. It is not always that if genotype is there, there has to be phenotype. Unless that genotype gets the proper environment, it will not be expressed properly. The phenotype will not be expressed properly. Like in case of Mendel, there were genes, there were alleles, the phenotype was there. But this is not true in all the cases. The responsible allele may be there in an individual, but the individual may not have that phenotype. Or if that phenotype is there, the magnitude of phenotype may vary. And that is what penetrance and expressivity means. So the penetrance is the number of individuals or the percentage of individuals which have the expected phenotype. Whereas expressivity is the level of expression, the degree to which a particular character is expressed. One very common example, polydactyly in humans. We know Hrithik Roshan, he has this condition. So this condition in humans is under the control of dominant gene, but it is not completely penetrant. And a survey was carried out in which 42 individuals were known who has the responsible allele for polydactyly, but not all. Only 38 persons had that polydactyl, then extra digit, either in form of finger or the toes. They were there. So it is 90% penetrant, right? And then comes the expressivity. This polydactyl condition can be at any magnitude. If you can see this lower left picture, it is just an outgrowth. If you see the middle one, yeah, it is something like thumb. But if you see this extreme right, you cannot tell which is the extra digit. So this level of expression, it varies. The allele is same. Its respective phenotype may be present, may not be present. Even if it is present, the degree of expression will vary. Then there are several examples, like in primrose, red and white flowers are there. White is homozygous recessive, red is dominant color. But this is expressed only if you grow the plants at low temperatures, below 20 degrees centigrade. But if you grow the plants at high temperatures, irrespective of the genotype, the flowers will be white. Whether it is homozygous dominant, heterozygous, or homozygous recessive, it will be white in color. Then another example of Himalayan rabbit. You can very well see the phenotype of the rabbit. The extremities are pigmented, rest of the body is white. So the Schulz, he said that probably the extreme parts of the body, they are cooler compared to the rest of the body. That's why the pigment is retained. It is not degraded. And it appears colored and the body appears white in color. So it has to be proved. So they have proved it experimentally by shaving the complete rabbit and rearing it at low temperature. When the rabbit was reared at low temperature, the complete rabbit was pigmented. In a reciprocal manner, they shaved the paws of the rabbit and covered it with a bandage. And after a few days, they opened the bandage. The fur was white in color. Why? Because this bandage, which covered the paws of that rabbit, it retained the heat which is dissipated by the body. And that's why the temperature in that region, we experience this. If you tie something on in some body part of your body, after some time it feels little bit warmer. Why? Because the heat is trapped in that bandage, and that heat caused the degradation of the pigment. And here the temperature is affecting the phenotype. Then this is the example of humidity affecting the phenotype. So Drosophila, you can see this. There are bands, chitinous banding on the body of that Drosophila fly. It is under the control of a sex-linked gene which is dominant. When the bees are reared at high humidity, under high humidity, irrespective of the genotype, the banding would be normal. But when you rear at low humidity, the homozygous recessive will result in the irregular banding, not the regular banding as you are seeing in this picture. So again, the environmental condition is affecting the phenotype. Then there are multiple alleles and the isoalleles. So as I have told you, alleles are just the alternate forms of the gene. And what is a gene? A sequence of nucleotide which codes for some RNA, transcriptional unit, as we call it. So there are several nucleotides in a gene, and those nucleotides, they have a certain sequence, and that sequence can change. Why it changes? It changes because of mutation. Mutation occurs. There are substitution mutation, deletion mutation, addition mutation, various sorts of mutation are there, you know. And this brings about the change in the nucleotide sequence. And each time when the mutation occurs, a new allele is formed. And that's why there is a wild type of allele, the original allele, it is mutated many times. That mutated copy is again remutated many times. And in this way, new alleles are evolved. And a series of alleles are there. And these are called as the multiple alleles. Among these multiple alleles, there are certain group of alleles which has the same phenotypic range. What is that? Like for example, here I have given in the bottom, there are three alleles: W plus S, W plus C, W plus G. These are the three alleles, different alleles, but their phenotypic expression is the same: red eye in Drosophila. So all these are called as isoalleles. Why? Because iso means similar. Their phenotypic expression is the same. That's why they are called as the isoalleles. And then there will be several alleles, and these alleles may be dominant with each other, may be co-dominant with each other. Like the classical example of ABA blood group. Three alleles are there. Out of those, two are dominant over IO, and they themselves, IA and IB, they are co-dominant over each other. So these multiple alleles can share any sort of dominant recessive relationship. There is no such rule that always mostly the mutated allele, the evolved allele will be recessive, but it is not always the case. It can be dominant also, it can be co-dominant also. So it depends. Then the most important is the gene interaction. Most of the phenotypes in an individual are under the control of more than one gene. And when this is the case, those genes, they interact with each other to produce a phenotype. And that interaction can be divided into two broad groups: epistatic and non-epistatic. Suppose in some during gene interaction, one gene suppresses the effect of another gene, you call it as the epistatic gene interaction. And then there are certain very few gene interaction when there is no such suppression or operation of the gene. They interact, they play with each other very friendly, in a very friendly manner, and produce new phenotypes. And this is called as the non-epistatic gene interaction. So a very classical example of plumage hen, or what you can say fowls. So here you can see this comb in the fowl, it is under the control of two genes, R and P. So this is the walnut, this is rose, this is P, and this is single. Rose is produced when the genotype is homozygous recessive P and either homozygous dominant R or heterozygous R. P is produced in opposite genotype, exactly opposite genotype, homozygous recessive R and coupled with homozygous dominant or heterozygous P. Single is produced when both the genes are in homozygous recessive condition, and walnut is produced when at least one dominant copy of R as well as P is present. So here neither R nor P is suppressing the expression of other gene, but rather when they both come together, they produce a altogether new phenotype, that is the walnut type of comb. As you can see this in cross, you cross a rose with red, P, you get walnut type. If you interbreed this walnut type, you will get 4 phenotypic classes: walnut, rose, P, single in 9:3:3:1 ratio. Then you come to the epistatic gene interaction. There are several sorts of epistatic gene interactions. You can see this. We cannot cover all this in this small lecture of 45 minutes or 60 minutes. I shall give you only one or two examples. This is the example of summer squash, a cucurbitaceous vegetable grown in hilly regions. Now, there are 3 types of fruits: white, yellow, and green. White is dominant to yellow and green. Yellow is dominant only to green, right? And that means green is recessive to both yellow as well as white. Green is obtained when 2 genes control this phenotype, W and Y. When both these genes are homozygous recessive, it is a green phenotype. When W is homozygous recessive with at least one dominant copy of Y, the phenotype is yellow, and white phenotype is produced under 2 conditions. There should be at least one dominant copy of W and Y, or one dominant copy of W coupled with homozygous recessive condition of Y. So here, this what you can say, presence of Y is the presence of W masks the expression of Y gene, which will at least then produce the yellow phenotype. Now, to understand this phenomena, let us see Okay, so first we shall see that crosses, where you cross a dominant, what that is the white colored plant with homozygous dominant W and Y genes crossed with green, which has homozygous recessive W and Y. The F1 will be heterozygous for W as well as Y, and it will be white in phenotype. If you self these plants, then you get 3 phenotypic classes rather than 4. Typically in Mendel, you get 14 phenotypic classes. But here you get 3, white, yellow, and green in 12:3:1 ratio. So to understand how this phenotype is produced, let's see the hypothetical biosynthetic pathway. The color is imparted by some pigment, right? So you know the biosynthetic pathway where there is one precursor, it is acted upon by some enzyme, it produces the product. That product is acted upon by a second enzyme, it produces the second product, which is again acted upon by the third enzyme, which again produces the third product. And in this way, a end product is produced at the end of that biosynthetic pathway. So this biosynthetic pathway starts with a white pigment. It will have a green pigment. It will convert it to a green pigment, and that green pigment will be converted to yellow pigment. So this is a two-step reaction. Pathway. First enzyme will convert white pigment into green. Second will convert green into yellow. So these genes W and Y, they play the role in these two steps. The gene W codes for some inhibitor, which will inhibit the enzyme which converts white pigment into green. Whereas Y will code for the enzyme which converts green pigment into the yellow. So what happens when the genotype is at least one dominant copy of W and Y? What will happen? There will be an inhibitor produced. The same will happen if there is one W coupled with homozygous recessive Y. The inhibitor is produced. It will block the enzyme, so the white pigment will not be converted to green. Now, under this condition, if the genotype is capital W, small w, capital Y, small y, this Y will produce the enzyme, functional enzyme, but it will be of no use. Why? Because the substrate of enzyme will be missing. And that's why even if the product of gene is functional, it will be not produce the yellow pigment, and that the phenotype will remain white. In another situation, when W is homozygous recessive and at least one dominant Y is there, since the genotype is small w, no inhibitor is produced, white pigment is converted to green pigment because enzyme is functional. Here, since there is one capital Y, at least one capital Y, the functional enzyme will be produced, and that green pigment will be converted to the yellow pigment, and the fruits will be yellow in color. In the third case, when the genotype is small w, I am sorry that this picture here shows capital Y. That is not the case. It is small w, small y, small y. Small w will produce no inhibitor, so white pigment will be converted to green. Since the genotype is small y, the functional enzyme will not be produced, and the green pigment will remain as such. It will not be converted into the yellow pigment. and that's why the phenotype of the fruit will be green. So this is one of the examples how the epistatic interactions they function in the and they produce the various effects. Mostly in textbooks you get the interactions of two genes, but it is not that there are only two genes to interact. There can be three also, there can be more than three also. And all those genes, as the number of genes increases, the expression becomes more complicated, more or less predictable. And then then there are quantitative traits like yield, plant height, and all those. And those are the polygenic characters which are under the control of several genes. Okay. And those do not follow the Mendelian laws of inheritance. Then the last phenomena of linkage. So Morgan was working with Drosophila. The wild type of phenotype is red eye color with normal wings. The mutated is white eye color with miniature wings. He crossed, and both these genes are present on X chromosome. They are sex-linked genes. So the females will have two X chromosomes, so two copies of both the genes. Males will have only one copy because they have only one X chromosome. So female small w with M plus genotype was crossed with a male whose genotype was W plus M. So obviously the progenies were WW plus M plus M. That means they had red eye and normal wing. The males, they were WM plus. They had white eye, normal wing. In another cross, Morgan crossed heterozygous females. They were W plus W and M plus M, means red eye, normal wings, but heterozygous condition were crossed with WM plus, white eye, normal wings. So the population was analyzed, the F1. The females were 50% white eye, normal wings, red eye, normal wings, and remaining 50, as expected. Males were of 4 different types. There should not have been 4 different types of males, only 2 different types of males. Because males will get one X chromosome from here and either this chromosome or this chromosome from the female side. And that's why there should have been only of 2 types. But here they were of 4 types. And what were those? White miniature, white normal, red miniature, and red normal. How these 4 male types of males appeared? So to understand this, you have to look into okay, before that, even if you consider all the probabilities, then each of them should have been in equal proportion. 25% white miniature, 25% white normal, 25% red miniature, 25% red normal. They should have been in that manner, but they were not so. Rather, white normal and red miniature, which were the parental combinations, they were more, 66.5%. And red normal, white miniature, they were less, 33.5. They were not in equal ratio. The ratio was distorted. Parental types were more, recombinants were less. How this happened? This is not according to the law of independent assortment, where each type of gamete is of equal frequency. But this is not the case. Then to understand this result, because by the time meiosis was known, crossing over was known, and much of these things were known, and that's why it was easier to explain these results. they looked into the meiosis of the females. This is the female composition. 2X chromosome, like this. So either a crossover will be formed between these two genes, or it will not be formed, right? So during prophase 1, you know that the crossing over takes place during Pecketin and all those things. And due to this, what happens? In most of the cells, there is no crossing over between these two genes. In few, there is. And even in those cells, at the time of meiosis, there are 4 chromatids. 2 chromatids in each homologue. Out of those 4, only 2 are under crossing over. Rest of the 2, they do not participate in crossing over. At least in the same, between the same loci. Mostly. So at the end of meiosis, you see that when there is no crossing over, all the 4, what you can say, cell, the products of meiosis, they have the parental genotype, WM plus, WM plus, W plus M, W plus M. Whereas if the crossover is formed, then the combination varies. WM plus, W plus M, WM, these are the recombinant combinations, right? So these are the parental combinations, and these are the recombinant combinations. And since recombinant combinations are lesser than the parental combinations, the distortion in that ratio was there. Parental were more, recombinants were less. And this, in this way, the Morgan proved that when the genes are present on 2 chromosomes, on the 2 genes are present on same chromosomes, they will not follow Mendel's laws of independent assortment. But then there is addition. When the 2 genes, they are present on same chromosome close to each other, you call them as completely linked, because such genes generally they inherit together for more than 2 generations. Whereas if the 2 genes present on same chromosomes are fairly apart, they are called as incompletely linked genes, because that linkage is broken within the 2 generation of breeding. But here we should take care that all the genes which are present on same chromosomes are not linked. There is another term for that called as syntenic. The genes which are present on same chromosomes, those are called as the syntenic genes. Like here, A, B, C, D, E, F, G, H, they are present on the same chromosomes. So A and H, they are syntenic. B and G, A and G, they are syntenic. They are very, very far apart. Whereas A and B can be called as linked, B and D can be called as linked. They are on the same chromosome. Like for example, A and say D, they are fairly apart, but they can be called as linked because their linkage cannot be very easily broken. Whereas the linkage of A and H can be very, very easily broken. So such genes are called as the syntenic genes. So there is just because the 2 genes are present on same chromosome, you cannot call them as a linked gene. They should be called as the syntenic genes, right? So this is how probably that's what I wanted to say. When Dr. Kirtane contacted me, she wanted me to speak something based on the Mendel's laws. Okay, because we are celebrating Mendel's birth anniversary. But if you look at the life of Mendel, the work of Mendel, Mendel is a monumental person. Mendel has divided this whole time period into two eras, pre-Mendelian, post-Mendelian. We call it pre-Mendelian genetics, post-Mendelian genetics. So that is the importance of Mendel in the field of science, as well, not only science, in the field of knowledge, or in the field of humanity. He has divided the whole time into two groups, pre-Mendelian and post-Mendelian. His work was the basis of genetics, modern genetics. But only those two laws is only the tip of iceberg. It was necessary because that work has prompted all other works. I have spoken only about the classical genetics. I didn't touch upon the molecular genetics. All those, if you look superficially, they appear to be unrelated to each other. But deep within, even the molecular genetics it has erupted from the Mendelian laws. Those were the two basic laws, or three, depending upon authors. Because Mendel didn't propose any law, he has only given the results and his conclusions. The later authors, they have divided his conclusions into 2 or 3 parts, and we call it them laws of inheritance. So based on those 2 or 3 laws, everything what we understand about genetics now is dependent, and that is the importance of Mendel. So with this, my tribute to Mendel, and once more I want to thank the management of Mulla Education Society and the principal and staff of Arts, Science and Commerce College, as well as the Yashwantrao Chauhan College, for inviting me, for giving me an opportunity to interact with you. So with this, I hand over the mic to the dais.
Thank you, sir.
Yeah.
Then I request to Laure sir, share feedback link in chat box, and all participants, please fill your valuable feedback. And then I invite Dr. G.G. Poddar sir, Head of Botany Department, Yashwantrao Chauhan College of Science, Karad, for vote of thanks.
Hello. Am I audible?
Yeah.
Okay. Thank you, madam. So I am here to express the vote of thanks for today's national webinar on the occasion of the 200th birth anniversary of Gregor Mendel. I am very much thankful to today's honorable speakers, Dr. B.J. Apparao, Director, Collegiate Education, Muda Education Society, Nevas Ahmednagar, and Dr. Rupesh Badere sir, Associate Professor, Department of Botany, Sant Tukadarji Maharaj, Nagpur, University Nagpur, for their excellent talk on the genetics. I am also thankful to convener of today's webinar, Principal Dr. S.L. Laure sir, Arts, Commerce, Science College, Sonai, for his convener speech. I am also thankful to today's President, Dr. S.S. Brungre, Head Department of Chemistry, Yashwantrao Chauhan College of Science, Karad, for his presidential address. I am also thankful to Dr. S.A. Kirtana madam for her welcome and introduction, as well as the organizing this program on the occasion of the birth anniversary. I'm also thankful to principal of our college, Dr. S.B. Kengar sir, for according the permission to conduct this program. I'm also thankful to Dr. Sandeep Khedkar and Mrs. PM Ghadge madam for the introduction of the today's guest speakers. I'm also thankful to Miss Snehal Patil for anchoring, and last but not least, the today's all active participants who have participated this national webinar. I am also thankful to all these participants for their participation. Thank you once again.
Thank you, sir. Thank you for your cooperation and can say suggestions, timely can say support from Arts, Commerce and Science College Sunai and behalf of Mula Society. I thank you all the participants and the speakers. Thank you.